So thanks, Andrea, for the chance to talk, and the team for putting this amazing symposium on. It's a highlight of the week for many of us. I'm going to talk today about a project that emanated from a talk I gave a couple of weeks ago, which is our attempt to understand what the genetic basis of variation in susceptibility to COVID-19 might be. It's an international team with myself and my group at Barnard, Juja and Zabox-Marker in the physics department, Imre Bartas at the University of California, Andy Munkatsky and his team in Victoria University of Wellington, New Zealand, Katsumi Higaki from Japan, Victoria University, and then two very skilled and renowned physicians, Mark Patterson at the Mayo Clinic and Mark Waldefang at the University of Melbourne, Australia. So why am I showing you a slide of sickle cell anemia on this slide here? Well, sickle cell anemia is the most classical advantage of a concept known as heterozygous advantage. Heterozygous advantage is a term in population genetics to explain why heterozygote has increased fitness relative to the other genotypes. And shown here is the classic sickle cell anemia profile of people that are homozygous to sickle cell anemia, dying at an early time point because of that disease, particularly relative to the control middle graph here. And yet people with heterozygous advantage, particularly in areas where malaria is endemic, have an advantage. The heterozygotes have an advantage over both of the homozygous genotypes. And that's because sickle cell anemia confers resistance to the malaria parasite. So what we would like to understand is whether heterozygous advantage in particularly lysosomal storage disorders might confer resistance or differing susceptibility to COVID-19. This sort of the organelle that I'm going to tell you about, a concept of what we will be seeing probably quite a lot of in our New York area, which is the accumulation of garbage and the need for recycling. And of course, the lysosome is the organelle originally identified by Deduve and colleagues that is responsible for degradation and recycling in the cell. Our studies commenced with the role of lipids, particularly cholesterol, in viral entry, viral infectivity, and later stages in the COVID-19 viral life cycle. But what I'm going to talk about mainly today is a more farther encompassing role of the lysosome in this process. And of course, the lysosome is the point of entry, not the point of entry, but the point at which the virus ends up in terms of infectious cycle, particularly the first stages of infection. And so I'll be talking about that in detail. But the bottom line is that envelope viruses need the lysosome, as do many other viruses. This is the lysosome. It's the acidic lumen of the cell. It's frequently disrupted by lysosomotrophic agents such as chloroquine and ammonium chloride. The proteomics have been developed very well. There are about 300 or so known proteins associated or integral to the lysosome. And it's well known that any defects or many defects in those proteins results in a lysosomal storage disorder, where metabolites accumulate within the lysosome and ultimately disrupt the function of that lysosome, of that organelle. And these proteins range from hydrolyses, which is the major component, major aspect of the phenotype or the function of the lysosome, to transporters such as the Mtc One Protein, which I'll tell you about a little bit as we go along. So lysosomal storage diseases were first identified back in the 60s, or characterized back in the 60s by Henry Gary Herz. And what he basically typified was the idea that pathological manifestations associated with lysosomal storage disorders accumulate because of accumulation of metabolites in that organelle. And that associates then with defects in the function of the lysosome. We started on this study because we were interested in the disease known as Niemann Pick type C disease, which is a lysosomal storage disorder associated with the accumulation of lipids in the lysosome. It's a monogenic neurodegenerative disease for which there's no current cure. So this is what we came up with, an idea I presented 2 weeks ago. Because the Niemann-Pick's critical role in cholesterol homeostasis, particularly in the lysosome, we postulated that if we inhibit NPC1, for which there are numerous agents that are capable of doing that, then we would affect the life cycle of the virus at 2 different points, or several different points, particularly in the first stages of infection and later stages of virus assembly and progression through the cell. Shown here is a classical staining of cholesterol accumulation in a Niemann Pick type C situation. Normal cells are shown here, where Philippine staining generally decorates the outside of the cell. In the absence of the Mtc One protein, cholesterol accumulates and other lipids accumulate in the lysosome, shown by this classical staining. And in a interesting demonstration of a pharmacological copy of an Mtc One mutant state, compounds such as this U1866A compound inhibit the Mtc One protein and produce the similar accumulation of cholesterol in the lysosome. So what we postulated was that if you intervene pharmacologically Mtc One, that would be an anti-infective agent. We showed several compounds that are FDA approved for a variety of different conventional uses that are very capable of inhibiting the Mtc One protein and giving resistance to a whole variety of different viruses. There are also compounds known as Mtc One mimetics, shown here on this slide, and those compounds have the same net effect in terms of accumulating lipid in the lysosome, but they work by a different mechanism other than just directly inhibiting the Mtc One protein. Nevertheless, the concept is the same, that changing this pathway, this lysosomal pathway, gives you resistance or reduced infectivity against a variety of viruses, including the coronaviruses. By way of an update, we're currently working with the NIAID to test these compounds. Many of them have already been tested for their effectiveness against COVID-19, and hopefully we'll have that data in its entirety fairly soon. By way of another update, this is the same assay I've described already, the Philippine staining of free cholesterol in the cells. Shown here is the Mtc One mutant, showing a characteristic accumulation of cholesterol. And here is the effect of chloroquine, a drug that's, and hydroxyquinoline is similar, a drug that's been touted as being a possible therapeutic agent. And here another interesting thing that we just recently got data on, that Katsumi Higaki got data on, is the role of progesterone in also phenocopying the Mtc One phenotype. Progesterone causes accumulation of cholesterol in the lysosome, perhaps raising the possibility that progesterone might be play a role in the gender bias that's been observed for COVID-19. This is the Mtc One protein. I talked about this before, but basically the Mtc One disease has a prevalence of between 1 in 90,000 and 130,000. Based on standard Heidi-Weinberg equilibrium measures, that would mean that there'd be a carrier frequency of about 1 in 180 individuals. Unfortunately, Mtc One shows no founder's effect, so it's not particularly represented in sequestered populations or populations that show that sort of phenotype. And so for that reason, we decided we should spread the net a bit wider and look at generally lysosomal storage disorders, because we would get more data and also aspects, investigate different aspects of biology. Imre Bartas came up with a modeling process, which he will talk about at the end of my talk, if there are any questions about it. Most importantly, this has been approved by the Barnard College IRB, and so we're already ready to collect data, and anybody who can give us advice on the best way to collect that data, we would gladly receive that advice. The reason that we decided to focus our studies on lysosomal storage disorders in general is that there are about 50 different syndromes that are associated with defects in the lysosomes. Their prevalence varies significantly. This is a study done in Australia that studied 27 different lysosomal storage disorders over a number of individuals. And they came up with a combined prevalence of about 1 in 7,700. Assuming, again, a Heidi-Weinberg equilibrium, that would give a carrier frequency of 1 in 42 individuals. So there should be a reasonable sample size there for us to study. The sample size might be even better in that when Fabry disease was studied, another lysosomal storage disorder, this one actually happens to be X-linked, but this had a, when this number of male neonates was screened for the disease, although the estimated incidence was 1 in 50,000, the incidence was much greater, 1 in 31,000. That would then give a carrier frequency of 1 in 28. So by combining these studies, by looking at lysosomal storage disorders in total, we should get some information about the population genetics of susceptibility to COVID-19. This slide, again, is a cholesterol accumulation piece of data that Katsumi actually got in the last couple of weeks. These are, again, the MPC1 mutant showing the classical cholesterol accumulation in the lysosome. And these are 3 different lysosomal storage disorders. I-cell disease, which is mucolipidosis 2, which is due to a defect in this hydrolase, this UDP-N acetylglucosamine 1 phosphotransferase. GM1 gangliosidosis is due to a defect in the enzyme in the lysosome that works on breaking down galactose, so beta-galactosidase. And Gaucher's disease is due to a defect in glucocerebrosidase. The reason I'm showing you this slide is to show that the distribution of lipids in these other lysosomal storage disorder is somewhat varied. In other words, I-cell disease looks very similar to MPC1 mutant because it accumulates cholesterol because of defects in the lysosome. GM1 gangliosidosis, however, shows an accumulation of GM1 gangliosides, but very little accumulation of cholesterol, although there is some there. And then Gaucher's disease, which is somewhere in between in terms of cholesterol accumulation. So by studying this whole set of lysosomal storage disorders, we're going to be able to mine into a lot of biology of the lysosome and investigate whether that organelle is critical for COVID-19. The other reason I showed these diseases is that they tend to show an increased preponderance and frequency in isolated populations. So Gaucher's disease is the most common genetic disorder found in persons of Ashkenazi Jewish ancestry. And I-cell disease is very common in Japanese populations, which was one reason for Katsumi to get involved with us in this study. The other aspect of this slide, which I want to emphasize, is that although we're assuming that some of these mutations will give resistance to COVID-19, they also could give increased susceptibility. And so the way that we're going to collect this data should enable us to unravel that question, we hope. So MPC1 deficiency is not just a neurodegenerative lipidosis. It is in the MPC1 protein has been shown to be involved in infectivity for filoviruses, particularly Ebola, coronaviruses, and also retroviruses. And so by studying the role of the lysosome in this infectious cycle, we think that we can actually investigate whether that organelle is critical for infection by COVID-19. Just to re-emphasize that this is a multinational effort. It was designed that way, both because we have different degrees of labs being open. So Katsumi is on the west coast of Japan, and at the moment he's able to go to work without any problem. Andy is in New Zealand and has just reopened almost freely in terms of doing his work in the lab, so he's up and running. And we're at a somewhat different stage at the moment in Barnard. Hopefully we will get to the same stage as has been observed with sickle cell anemia in MPC1 disease. It's been a very important collaboration with family foundations. So the National Niemann Pick Disease Foundation and the International Niemann Pick Disease Alliance have indicated their willingness to collaborate with us in collecting this data, at least for Niemann Pick disease. And we're currently investigating other family foundations to acquire that data. We're going to use a questionnaire. That's great. I'm almost finished. We're going to collect this data by, in the first instance, questionnaires. That questionnaire has been approved by the Barnard IRB, so we're very much ready to go. I'll leave you with this slide, which shows the heterozygous advantage. In our dreams, we would see some association with a lysosomal storage disorder, showing heterozygous advantage for that lysosomal storage disorder. Most of these diseases are autosomal recessive, and most of them are true autosomal recessive in that they have no penetrance for the disease in the heterozygous state. And so we'd be very interested in finding out if we can get something that looks like this for COVID-19. And of course, then that directly feeds into the idea that that gene that is defective in the lysosomal storage disorder could be a target for therapeutic intervention. Thanks for listening. Happy to take questions.
Thank you, Steve. We have time for a few questions. I don't see any raised hands.
Hi, this is Neville. Can you hear me?
Yep.
Yeah, Neville, go ahead.
Yeah, I was wondering if you could comment, given your expertise on the pathophysiology with the chloroquine or hydroxychloroquine and the lysosomes, whether the use of those drugs, in your opinion, very early in the disease or even as a prophylactic has a much greater chance of working or not compared to later?
Well, you know, from a purely lipocentric perspective, which is where I come from, I would agree with you. I think that these drugs, because they are lysosomal trophic agents, would affect that first stage of infection with the disease, and so therefore could well be some sort of prophylactic. Obviously, in the absence of true clinical trials, that's what the bar needs to be. But that would be my bias. But of course, those drugs have other effects on the cell, and so it's not clear whether whether what I'm describing is the primary mechanism of action, or whether all the anti-inflammatory aspects of those drugs is important as well. But we'll certainly be investigating that, particularly in comparison to what we're looking at.